So this is I call this advanced concepts of DFT, higher dimensions. So it goes toward cognition, but it's still very elementary in some other way. So we already talked briefly about higher dimensions because I think someone asked in the early lectures about that, and I stated then that yes, we can look at fields in multiple dimensions, and in the CEDAR tutorial you played with that. So we've used the one-dimensional fields most of the time, and you already saw two-dimensional fields. We routinely use three-dimensional fields. We have used four-dimensional under some special circumstances. We use a lot of zero-dimensional fields, which are just nodes, individual nodes that play a role. So I earlier argued there is nothing special about these higher dimensions, even though mathematicians like to worry about that. But certainly the attractors and the instabilities that we use exist there. The higher you go, the more exotic solutions you find, and those might be artifacts or might not be nearly relevant. Unless some new discovery is made. So that's not what I'm talking about. I'm talking about how higher dimensionality will lead to new functions, and that comes from the question of what these dimensions are. So the interesting stuff happens when we're combining different things, like we're combining something like color and visual space, right? These are very different dimensions. Mathematicians always say that's a space, right? But a physicist, you would say, well, it's meters along one axis, and it's, I don't know what the color units are called, I forgot that. Hue values, along another, or orientation would be degrees in one direction and centimeters along another dimension. You can already see that it's a bit superficial to say it's just one space. It is not even clear if there's a good metric in that space, right? And so that's where you really want to look carefully for what it actually means to have these peaks. And so that's what I want to take you through. And the first thing I want to address is what is called binding. So here I have a simple toy example. This is from the 5th chapter of the book. I always will use for a couple of slides now a visual space that I'll only track in one dimension. Take one spatial dimension just so that the plots are two-dimensional. In the other case, it will be then three-dimensional because visual space has two dimensions and color will be added as a third. So one-dimensional space, and then there is color in the scene. And so I will be talking about this two-dimensional space that has color on one axis and spatial location on the other axis as a joint representation of visual space in the sense of retina space and color. Joint in the sense that neurons in there are tuned to visual space and tuned to color. You know that the color actually in the brain has these opponency systems and somewhat special kind of tuning, but we always use color because it's kind of very easy to intuit. It's maybe neurally more clear about orientation, like orientation of little edges, where you have neurons that have these tuning curves that I showed, the Hubel and Wiesel tuning curves. So they have a receptive field. That means they're tuned to space. They only respond in a certain spatial region. And they're also tuned to orientation. So they are they're representing both of these dimensions, right? So we talk about these being bound, and you will see, as I explained, that why we even use that jargon. So they're represented jointly. And that means if we think of this 2D field, then that would be an abstraction of the corresponding feature map in cortex. In visual cortex, you have populations of neurons tuned to 2 or 3 dimensions like that. And if you look at the ensemble of these firing, the population code, then it would be modeled here by this field. And that means that the visual input to those cells is two-dimensional, right? For instance, this little element is located and has a color, and that would create a 2D input that creates maybe here a peak at that location, and the same here for that object. Now, I'll show now how you can get stuff in and out of a bound representation like that. So for instance, you can extract the features that are bound and isolate them. It's very simple. If I have a one-dimensional color representation, so a field defined of a hue, then I could just sum across all spatial locations and provide input to that field. I could sum by just summing and taking a threshold or their softmax, different, whether I preserve how much activation there was. but it doesn't really matter that that will be how I extract that information. This is sort of like marginalizing a probability distribution. If you know the probability density, you have two variables, and by summing over one, people say marginalize. That is, you assume that you have certainty about that one dimension and don't need to keep track of its probability. And very similar, I can just sum along the Hue axis and extract the spatial locations. Right? So if you have bound representations, you can extract the components from that. Right? It's almost trivial, but just want to make that point. And these kind of coupling structures are actually available in CEDA. I'm not even sure, it was maybe even already explained. You can also do the reverse. That is, you can assemble not unbound representations and bind them. And that is done by ridge input. So ridge input would mean, for instance, if you know the color, then you can say all the neurons that represent that color get some input, irrespective of what spatial location they encode. And similarly, you know, if you know the location, all the neurons that are tuned to that location, but irrespective of the color they represent, are being activated. No? So ridge input is a very simple form of coupling. And you can arrange things so that only where these ridges overlap does the field go through the detection instability. So a peak will form without any visual input, right? Now will form just from these two inputs wherever these two superpose. There will be a question of the threshold. You can make sure that that's actually happening. And so that's how you would bind to previously unbound representations. And you will see in some representations that's a tool that we can use. That leads to the binding problem. Some of you might have heard about the binding problem. It's discussed also in other frameworks of how you go about binding. It's a concept in cognitive psychology. So the binding problem arises here in the sense that if you are trying to assemble the features of two objects at the same time, for instance, you know, there is a blue object and a green object at these two locations. If you just put in these ridges, then there are two possibilities of combination. You don't know if the blue is at this location or is it at that other location, right? So it leads to what people call illusory conjunctions. There are some special conditions under which humans actually suffer from such mistakes, and we modeled that. So Sebastian Schneegans did. But generally, we don't suffer from that in general. And the answer is that you need to assemble objects from individual features by looking at one object at a time, because you only have that when there are more than one object, right? If you have a single object, works fine. And in fact, that is in our theory and also consistent with older theories is one origin of the sequential processing bottleneck. So when you look at a lot of cognitive operations, it's always one object at a time. There's a lot of evidence for that. For instance, psychological refractariness, some of you might know that, or attentional blindness, the attentional blink, and then the characteristics of visual search that Raoul Greben will be talking about. These are as evidence consistent with that kind of thought. Now, when you have these two operations, you can combine them to do visual search. So visual search would mean that, for instance, here you have a scene, there are two objects in the scene, and you know that you're looking for the blue one, and that knowledge is expressed as having a peak in your blue field. That will be a Q that you have. And then you feed that peak in, and you can arrange, again, through control of threshold, of resting level, can arrange that only where a two-dimensional input from the scene overlaps with that ridge, namely here, a peak forms, and not where it doesn't overlap. And as a result, a blue item in the scene will create a peak, and you can read out its peak location, and then now you have found it. Now you can send that spatial location to your arm or your gaze and so on, and then work on it, right? And that's very fundamental step for almost all action and cognition, is to bring an object into the attentional foreground. And so Raoul will be reviewing a model that's fairly complex because it does a lot of things, but at its core is this mechanism. And he will not talk about ridges. He will talk about slices, because visual space is generally two-dimensional. And so if you know about the location, then you're actually depends on what. No, sorry, it's two-dimensional, exactly. And bringing in one particular color value then activates all 2D locations consistent with their color value. So that's a slice rather than a ridge. I just use this term to make it concrete. So just this opposite of marginalization. Okay, so that's a core use of the binding issue. So this form of binding is, most people don't even call that binding, because it is, in a sense, it's neuroanatomical, right? You have a neuron for every combination, right? In this field, neurons for every combination of these values exist. So that's not a very universal thing. I think I have a slide here that estimates that. So for instance, if you were just to try to bind orientation, color, texture, scale, and two-dimensional visual space, if I counted correctly, these are 6 dimensions. If you only wanted to bind those, or have neurons that are tuned to all of those things and all the combinations, and you use 100 neurons per dimension, then you already have 10 to the power of 12 neurons, which is the entire human brain. I took that, the numbers exactly to come out to that. So it's clearly something that doesn't scale very well. We humans have, according to psychophysicists and neurophysiologists, dozens of dimensions like that, visual feature dimensions. The exact number is debatable, but certainly not a very small number. It's also not like in the thousands, not even in the hundreds. It's probably in the dozens, like 30 or 20 or 30. But in any case, it certainly cannot be represented in this anatomically bound form. And that's where you now see the reason why we talk about that as binding. So the idea would be that you anatomically bind features only in low-dimensional subspaces. So this is what I was earlier hinting at, and what Raoul Greben will be referring to in his talk, that we typically think of neurons always having a spatial dimension. That is, all neurons have spatial receptive fields. If you go very high in cortex, for instance, to IT, you find that these receptive fields are large. They can be sometimes like half the visual array, but not the whole visual array. So they are localized, and they have a profile. So they're still spatially structured, right? They're not equally sensitive anywhere in the visual array. So that's a notion that you share the dimension of space. That's why I'm plotting this to be aligned along space, X and Y, that reflects retinal space, let's say. But that then actually are tuned to only a small number of feature dimensions. And we don't, of course, know experimentally also exactly which ones. And there could be combinations of features that we're trying an experiment. And it could be that they are maybe tuned to more than just one. Maybe they're tuned to 2, or maybe sometimes to 3, but a small number of those. And then binding, in the sense of knowing all the feature information about a given object, would take place through space. The notion is that you want to bind features because they pertain to the same visual object, and the visual object has a definite location in the visual array. And so that it's an appropriate thing to bind through space. And that goes back to Anne Treisman, who introduced something called feature integration theory that went through a couple of iterations. And there's good psychophysical evidence for that. The way she framed that, and people working around that sort of fold, wasn't actually really neural. It was sort of using that a feature file where you store features in a file and so on. And so one could say that dynamic neural field theory provides a neural account for binding through space. I think I have slides here. I'll briefly take you through that. I should say there is a theoretical alternative to that is also being promoted by some people, who say that you have a special agent for binding. It's not space. It's something like frequency. You have neural oscillators encoding these things, and that the phase of these oscillators represents whether 2 such oscillators belong to the same object. This theory was, in important ways, developed by Christoph von der Marzburg, who was my colleague in Bochum until a few years ago. He's now an emeritus professor in Frankfurt. and I'm not sure he still promotes this very strongly, but there are a couple of people who argue for that theory. We have argued with them. I believe that we have refuted it, but you can never know. There could be some maybe some special stuff that we don't know yet. So it's not the only possibility of how to do that neurally. And there's some people who really still commit to the oscillatory picture, even a colleague in Bochum, Markus Werning, for instance. So just a little bit more about that. So the idea of how to do the binding through space, see, this is the ridge picture I was talking about. If you have a representation of size, orientation, and color always over space, and you know the color, then you can express that by just putting a ridge through the field, and then only locations where there is input along that ridge would light up, and similar for the other feature dimensions. And binding through space could occur by saying, if one of those layers has a hit, so there is something with the right color, then there is some spatial coupling among the different fields where all the locations in the other fields that match that hit would also be activated. That can take place through a shared input, through a salience field, or it can be mutual activation. There are different variants and different models, but there's always this idea that you then favor selection of feature space representations at the same location. Here's an example of that from the book. Let's say you have this somewhat a toy task. You have the stimulus, and you have the task to say what is the green letter in there. Then we're assuming here you have a feature for color space representation, and then the shape is just here abstractly some feature. We haven't exactly how you represent that, and so there would be different shapes as a function of retinal position. So these little blobs are the 2D inputs into the system, and so knowing that you're looking for the green means you're putting some peak into your green field that gives a ridge input to this. The ridge input creates a peak at the matching location, and that is read out into a spatial representation. That spatial representation feeds back into these two fields. And so a moment later in time, this feedback has led to a peak in the second field at the matching location. You see this location matches, and therefore it is driven here by the spatial peak. And that is read out to the shape field, and therefore a peak reflecting the shape at this location comes up. You can answer it's an S. No, the green thing is an S. You see there's also a ridge here, because all of these connections are bi-directional, because you could just as well ask, where is the S? And then you would put a ridge in here, and then it would bring out the Q. So that bi-directional coupling is always present, and the whole thing is actually one closed loop dynamical system. So that would be sort of how binding would function in practice. So Sebastian Schneekans has worked out things like that, and I just hint here at that in the chapter 8 of the book. It's about how bind representations then are in working memory. I'll mention that because in a moment I will talk about an important wrinkle of that. So let's say you have this visual representation, a visual scene, where again, I'm using only one dimensions for the retinal space here for simplicity. And let's say you have these three objects that have color features and orientation features. Here is color, here's orientation. So we represent them separately by two feature space fields. And so the scene here would make these three colors and these three orientations was arranged to have this regular structure. And let's say what we want to do is we want to build a working memory of that scene. That will be here on the right, a working memory for the same feature values, where we store these three peaks as three sustained peaks that will survive the removal of the input. We will actually want that working memory to be in a different reference frame, namely in the scene reference frame, so that it is invariant under gaze shift. So we're looking around, the scene doesn't change. In fact, if you look around, your impression is that there is the world out there, even though on your retina you have this wildly changing stuff. Every 300 milliseconds your retinal input is totally changed. And so the basis on which you look at scenes is actually not your retinal image. It's a invariant representation of the world that somehow must, get rid of that variation. And I will, in a moment, talk about how such coordinate transforms are done. So we want to enter this here into this representation, and you see that there is so I'll explain now how we can do that by extracting the features and then binding again. And then in a moment, you will see why we do that way. When if we were to, let me just say, if we were to try to directly copy this over here, it would mean that we have to transform this entire space. It would be this whole column and coordinate transform that. And that, you will see in a moment, would actually be, again, extremely costly. It would take more than the brain to do that. And so instead, we're doing that only once. We're making the coordinate transform at a purely spatial level. We're just saying where stuff on the retina is mapped in the scene. And then we assume that we know the location here corresponds to that location there. And then we will transform, extract the features and assemble them again. So here's a little sketch how that works. For instance, we would first select, you have to do it sequentially, right? If not, we will have misbindings. So we select an object in a similar as Raul will explain. And that selected object, we know where it is in the outer space by the coordinate transform. So we put a ridge into this attentional field here in the scene space. Here in the plots it's at the same location, but it could be shifted. And so we extract the color of that feature that was attended, put that in as a ridge, and there we've assembled it. The difference between these two fields is just this has multiple peaks sustained, and this is only one peak for selection. So that's just technicality that one could do that in one field through more complex neurodynamics. And at the same time, this oriented object is selected, its feature is extracted, put in here, and get the result, right? And then next thing is you select the next item, inhibit the previous item, and you know where that item goes, and do the same sort of thing. And in the meantime, this is actually, in the meantime, the other one has already been put there, and so at the end you have that representation, and those are bound. Work came about by being bound in space, and you see a little bit the risk of misbindings. That's where the illusory conjunctions come from, because these interactions along this axis and along this axis are present, right? Because they are not turned off once we put it in. There's always this resonance coupling back and forth. And that's why there are these locations where there is a little bit of a risk to maybe by a fluctuation get an activation. And Sebastian Schneegans in this chapter shows how you can account for change detection results, where people have to look at a scene and have to memorize that scene. You show the scene maybe with one item changed and have to see if they detect that. And you can probe whether they are, how sensitive they are to misbindings, to confusing an object where the same features are present, but they are combined differently in different objects. And that can be account for these kind of results. Good. So to go deeper into that, it requires, it's something that just read the chapter and you will find that maybe very interesting. Good. That leads me to coordinate transforms. Coordinate transforms are actually, in some sense, analogous to binding. And that might be something initially perhaps surprising. And that's what the next thing I want to explain. So first, I want to point out that coordinate transforms are fundamental in sensorimotor forms of cognition, and I'll use eye movements here to explain that in a moment. But they're also actually important in mental operations. I'll briefly hint at that, and you will see that in the presentations of Mattis and of I think Daniel, probably at least those two. So let's first talk about what that actually is. Let's take the visual example. This is from Sebastian's chapter in the book. You have a visual scene, these two objects, you look at it with an eye, you have this on your retina, you shift your gaze on looking at this object. Now here the red is in the fovea in the middle, and now this green one is in the fovea, so the visual image has changed, although the visual scene is of course invariant. So the retinal frame is variant, and the coordinate transform issue is that you might want to have a representation of the visual space that is invariant under that gaze shift, just like the visual scene is. For instance, that is anchored, let's say, in the body or in the head. I'm sort of not looking into difference between eye, gaze, and with the head, sort of mingling that together, which is neurophysiologically correct. And so the other thing is really the body. And the arm is attached to the body, so for reaching, it's important where the body is. You don't have an arm attached to your retina, right? That's exactly the problem. And so you need to extract the information that is invariant under gaze. You can reach for an object and look at different locations in space and still reach correctly. So that's a very concrete way of talking about the coordinate transforms. And this shows up in a lot of places. I mean, this is what I just explained about being able to reach also when you look elsewhere. I earlier referred to the fact that your movements go straight to a target. So when you does that, you must be representing this not with respect to your head or body, but with respect to where your hand is, right? If you move your hand around, the movement parameters vary, and the coordinate transform would capture exactly that. And this is an example of this higher level stuff that Matthias Richter will be talking about. For instance, you might think that in your brain you have only a limited set of neurons that are tuned to something like to the left of or to the right of. So for instance, these neurons may be attached to some spatial representation where everything here in this left corner is voting for left, and on the right they vote for right. And now you want to apply that to just any visual array where the target is the vertical, the reference is the horizontal, and you want to say this is to the left of that. The vertex is to the left of the horizontal. And you want to be able to do that irrespective of where this reference is. So if you vary that along space, you want to be able to apply this operator to that location. Now, only two possibilities. Either you have a copy of that operator I call this operator, this little neural network here. You have a copy of that neural network for every retinal location, as some of the deep learning models assume. Or you just have a small population for that, and now you have to bring the activation from the sensory surface to that neural network so that it is centered on the reference object. So you want to, in other words, represent the target in a reference frame that is centered in the reference object. And that's the solution that Matthias Richter will be showing. So why is that a problem? Coordinate transforms, you can write them down on a piece of paper. For similar case, you can write down a formula for that, and that's what we did on that robot. When yesterday, I think I showed that we just took the coordinates on the robot body, and then knowing the heading direction, we were able to compute where it is in the world, and was just adding two numbers. Very simple. And for the retina, it might be with a sinus and cosines, but also very simple formula. But that's exactly what the nervous system is actually not good at function evaluation like that. And it is good. The nervous system can be described as function evaluators in the feed-forward neural network sense. I mentioned that at the outset. That is, if you have one representation like the retinal one, and you project onto another one, then you can say this is the output of that, and the red arrow would be the neural network, that has some structure to do that. So that's okay, that could be done. But a coordinate transform is something a little bit more complex, because it is a mapping that depends on some other thing. We call that steered. So for instance, the mapping from the retina to the body frame depends on where your gaze is. Or the mapping from your body space into the hand center space depends on your hand position. Or the mapping from the visual array to the object-centered array depends on the position in the visual array of the reference object. So we call that a steerable map, and that is not something that feed-forward neural networks do in a straightforward way. In other words, it's only fake function. It's a fake way to talk about these as being function evaluators, because there's only one fixed function. It's not a function with multiple arguments, right? And so how can we do that neurally? And the answer to that is that you need binding. And what you need to do is you need to represent everything that you want to transform and everything that transform depends on in a joint space. Then you come back to a feed-forward picture, right? Then you can use a mapping onto a third space to do what you want to do. But that's not enough, because when you have bound that representation, you also need to be able to now select in that representation which of the subset you're going to currently use. And I'll show you how that goes. So the bound representation is essentially just a higher dimensional field that has as one dimension the steering variable, and as the other dimension whatever you want to transform. So going back to this eye movement case, we will think of the thing we want to transform being the retinal space, and the steering dimension being the gaze direction. And for now, I'll just do both of those one-dimensional. We'll come back to the issue later. So let's here on the right, we have a human looking here straight ahead. There's a object a little bit to the right. The stimulus will always be at the same location in the body space. So this here on the right, the body-centered representation will be invariant across multiple pictures I'll be looking at. And the retinal location is 10 degrees here. You see this peak is at 10 degrees, and the gaze is straight ahead at 0. So we have a ridge at 10, a 10-degree reference object, and therefore we will get input into this bound field. With these two ridges, they overlap here, and you generate an item at this location. So it doesn't make sense to assume that there would be a direct 2D input, right? Because it's very different systems, visual array and the other gaze. The estimate of gaze comes from some sensors in the eye, that tell you where your eye is. It's actually for body frames also head neck sensors that play a role, and there are actually subtle calibration processes that use vision even for that, because you can. Anyway, let's not go there. So that's your joint representation. Now, if we have a slightly different gaze, so we look directly at the object. Now your retinal location is 0. Your gaze has shifted to 10. You get the peak here. This is where the previous peak was. Still same scene. And here's another one. You look to the left. You have now shifted your gaze to plus 30, and sorry, to minus 20. And on the retina, it's at plus 30 then to the right. And then you have that location. You can sort of guess what happens. It's along this line that the visual stimulus will fall in this combined field when the visual array is constant. And so all that you need is to project out, marginalizing again. All the locations here will contribute to a single peak. And if you do that on different diagonals, if you figure that out, you will find the body-centered position of the visual object. Right? And of course, it could be, this is just to make it intuitive, it could be a more complex thing. You could have a curved line and so on. If you need to represent more complex functions, it will be just a question of how you do the marginalizing here. Okay, so that would do the coordinate transform for you, and it turns out that that is something that is neurophysiologically real. So there are neurons that behave like this transformation field in an area called LIP in parietal cortex. These are called the gain field neurons. They have, see, if you measure the neural firing as a function of where this retinal stimulus is, you will measure its tuning curve. And if you move the gaze through, what you will find is that the tuning curve will be gained up or down depending on whether the ridge falls onto that location or not. And that property has actually been observed in this area. It is quite possible that there are more complex things like that, because a lot of the neurophysiology, functional neurophysiology like that is very, very demanding. You have to have animals who are trained, awake, behaving, and do a year of training, you operate, and then you have one question you can ask. And so a lot of experiments have not seriously probed this invariance by not actually allowing the monkeys to shift the gaze or to move the head. And it's quite possible that a lot of things that we think of being retinally based or being in a particular reference frame might actually be much more invariant like this. There is a story about, for instance, whether a colliculus is in saccade coordinates, which is retinal, or is in gaze coordinates. And David Sparks, who was a cheerleader of that entire approach, actually started to be able to record from head-free animals, and then found that a lot of the stuff that they used to think was saccadic coordinates was actually gaze coordinates. So things like that are possible. So this is neurophysiologically real. We believe it is still rare in the brain. There might be only 1 or 2 systems like that in the entire brain. And I'll give you some arguments for that in a moment. Just want to point out that these couplings we will typically assume are bidirectional. They would be naturally bidirectional if they are learned through Hebbian learning, because they just pick up covariation. And the bidirectionality actually gives you a different function. You can imagine that this would allow you to predict where the retinal input is. You know, if you know where it's in your body or head frame, you could predict where it goes when you change gaze. And that is actually something real. This is the same picture, and just points out that the mapping can handle whole arrays. It doesn't have to do just one peak at a time. You have to have a unique, one of those axes has to have a single peak. If not, you get the binding problem. It's enough for the gaze field, and you only have one gaze estimate, right? You don't split your eye into 2. So this one will be one-dimensional, and then you can have any complex landscape here and there. And this actually is, I'll just show you the movie. This is an example where you put in a retinal stimulus, you know where it's on the body. Now you extinguish the stimulus, make a gaze shift, and so you're predicting where on the retina the stimulus would be if you that you earlier saw. And that turns out something, again, that is neurophysiologically real. This is work, I should have written this down on this slide. It's, I forgot the name of a famous neurophysiologist who found that, though. This is drawn after their experiments. It's in IT, in the inferotemporal area. And I just forgot the name, sorry. So it's very intriguing. You have neurons there. The monkey fixates here, and this is the receptive field of neurons in this area. So if you shine a stimulus into this area, this would be that stimulus, then the neuron fires. In fact, when you remove the stimulus, it keeps firing for a while. It's like a working memory. But if the monkey makes a saccade that moves the receptive field out of that stimulated area, then the firing goes down. And then here's the amazing thing. If you're fixating here, you shine a stimulus into this location. This is the short little stimulus. And then the monkey makes a saccade. So now it falls into the receptive field of that neuron. The neuron fires, even though it has no input. It fires essentially based on prediction that there should be that stimulus. And then it goes off because the stimulus was removed. That's amazing, isn't it? And that the model that I hinted at can model that sort of thing. Right. And there are more things like that I want to skip, and end by saying, these are the coordinate transforms are costly. So one thing you can see immediately is that it's already more complex because the visual space is 2-dimensional, of course, right? So you have a 2-dimensional receptive visual array, and gaze is 2 variables, and then your body frame is going to be 2-dimensional. So the transformation field is a 2x2, so a 4-dimensional field, right? And this is Sebastian who sketched this here by a mosaic of 2D, a 2D pattern of 2D fields is a 4-dimensional space. So if you were to transform, so that is just space to space, and we're concretely hypothesizing that that field exists and that you're making that transform. If you were to make that transform together with the feature values, like you have now red over space, you would have to have that third axis also here, right? Because you have to keep track of whether it's the red thing that was at that location or the blue thing at that location, and also goes here. And similar, if you had, anything else you add, always is added to that. So you can see how that would scale quite badly. The dimensionality of this is always, has 2 more than the dimension of the visual array. And that is one argument why we don't think that the coordinate transforms would be done directly. And if they're not done directly, that is, if you have the disassembled objects, you would be able to do it directly only if you combine all the feature values in one representation, and transforming that would be a nightmare. So you don't do that, and then you have the binding problem that I hinted at earlier, and that's why you need to do this one object at a time, as in the transformational working memory that I showed you. Okay, I don't know if you can follow that so quickly. Quite challenging, right? So this is just the text for that. So we believe that the binding problem with the coordinate transform problem really are the origin of the sequentiality bottleneck that goes through all human cognition. It's kind of amazing. That is a thought, and the thought is, and you will see this emerge over the next 2 days, is that even mental operations are like that. When you think about a scene or think about an argument, you're making a mental map, and in that map you're associating particular feature values or properties of thoughts and actions with locations in space, and you're able to transform those. And to do that, you have to do one at a time for exactly these reasons here. So that's quite a fundamental constraint, right? Computers can do that differently. So I think maybe the last point I make in this lecture, and hopefully we're not totally off time, is to address that I was already talking about architectures without really much to do, right? I mean, these are all architectures, right? There is a field here, coupled, and so on. And I was saying peak, detection instability, selection, and so on. So why is that possible? So let's, why are, when you couple dynamical variables, things happen, right? It's like changing the equation. There are new terms in the equation. Why can I still use our concepts of the instabilities, detection, and so on for these architectures, for the individual components of the architectures? This is not a trivial problem. This is actually the original problem of analog computing as compared to digital computing. None of you will remember that because this is way before most of our lifetimes. Early days of analog computing was not anywhere near as successful as digital computing, because analog computers are very difficult to program. They're difficult to program because if you have analog computer is a big nonlinear dynamical system, and if you just add one term to that, everything can change. And therefore, they require complete mathematical analysis. And the digital computer is exactly the solution to that, where you isolate the individual elements. They're just flip-flops. And by the fact that they have this bistable dynamics that is very robust, you can now couple them, and they are still individually described as flip-flops. And that makes them invariant. So you can add a flip-flop that doesn't change the physical dynamics of everyone else. And the normalization that is achieved in silicon is exactly that the voltage that you have isn't affected by some transistor somewhere far. So in the brain, that's not immediately obvious that we have something like that in the brain. Well, you could think about the spiking mechanism perhaps helping with that. And so at our level of abstraction, it is this fact that we're dealing with attractors that gives this system the structural stability. Structural stability means that if you change the equation a little bit, the solutions are still the same, especially the attractor solutions are still the same. And it's a property of mathematically, when you have an attractor that is also structurally stable, there will be parametrically a neighborhood in parameter of that attractor where you still have a matching attractor, which could be, you know, shifted a little bit, but it would be still have the same layout. And in fact, you can also apply that argument to bifurcations. You know, through a bifurcation, attractor is not stable, of course, because, you know, it's lost, but you can include the parameter change as part of the dynamical system. Mathematicians do that in some funny way. It's like a marginally stable variable. And then you can say the whole bifurcation diagram is structurally stable. And that is actually the reason why we can do this. That is, when we couple the fields into architectures, the attractor states make that, as long as that coupling is, you know, sufficiently weak, the individual dynamics are still have the same structure, that is the same attractors and repellers and instabilities. And in fact, when the coupling becomes strong, the first thing that happens is that we're going through one of those instabilities. So we're saying one field pushes the other through the instability and so on. So we can still talk about the architecture with our concepts. And I want to show you one example of that nature. That doesn't mean that you could write, of course, an architecture where something wild happens, if you're outside the regime that is well described by our solutions and attractors. So when you build CEDAR architectures, you can get, sometimes you can get oscillations, or everything goes and so on. So that's the tuning, to stay close to these core solutions. This is not very difficult per se, but, you know, if you have specific demands like working memory and selective, or what the capacity of working memory is, then yes, there can be that's a real constraint on your dynamics. So here's an example of this that illustrates that the dynamic field architectures are not only robust, but they have some hidden flexibility. Matthias Richter, I'm using an example that Matthias Richter will be elaborating, and Daniel Sabinas will be actually pushing that further. So it's about spatial language. I'll not explain the whole story because he will, but I just want to use this example to sort of show actually two aspects about the architecture. So it's an architecture about spatial relations. So for instance, you want to say the green is to the right of the red, let's say. The green is to the right of the red. In a phrase like that, the green is the target, and we have a spatial representation for targets here, and the red is the reference. We have a spatial representation for reference here. The green to the left of to the right of the red. Now, green is target, red is reference. Now, general thing, if you say the ball hits the goal, you know, then the ball is the target, and the goal is the reference. And a lot of verbs and relations can be viewed that way. So here is a scene representation, is a visual feature field, a three-dimensional field with color and space. Just three slices through that field are plotted: the red slice, the green slice, and the blue slice. And this is an instance of doing the visual search. So by giving some boost to the red slice, you bring out this layer can make a peak, and it feeds it into this target layer through a forward connection. But this connection and the connection to the reference field have the same strength. It's plotted in bold just to say where it actually happens. And the reason why this goes into the target field is, at the moment when we're doing this boost, we're also boosting this. That is, we're giving a constant homogeneous input to the target field, but not to the reference field. And that makes that the target field can go through a detection instability with the input it gets from this layer, while the reference field can't. Once we've done that, we're telling the system to look at the red as a reference. We put the slice into the red, and we boost the reference field. Now only the reference field can go through detection instability, builds a peak at that matching location, and the target field, you know, gets a little bit of input, but it's not boosted, and therefore this is not enough to build a peak, and therefore it's not enough to delete the working memory of that previous peak. So what you see, through this boost method, what we're doing with this boost is we're controlling how close activation inputs in a field are to the detection instability, whether or not the field is ready to make a peak. And the effective input is making peaks, going through detection. That's when you, in effect, have achieved something. So in that sense, we can modulate the input strength to the field through just the boost. And that's, again, how the bias term, the homogeneous input term, that's also used in the memory trace, plays such a more important role in our thinking through the detection instability than, let's say, in conventional neural network architectures that don't use these instabilities so strongly. So in conventional neural architectures, this is done by gauging, which is some multiplicative role on the connection. So if you want to, if you have multiple connections, you want to select one, you multiply that one with a non-zero number and multiply the other ones with 0, which is called shunting. And that's how you can get flexibility. And for instance, there's a whole theory by someone called O'Reilly, who have kind of a lot of things with these shunting and gating mechanisms. And essentially, I'm saying that in dynamical field theory, that comes for free. You get this kind of gating stuff. Good. So that is as much as I wanted to say about higher dimensional fields. So they enable cognitive functions, new cognitive functions through binding, attention selection, matching, visual search, coordinates forms, which I showed, and through the robustness of the stability of the solutions that we give meaning, we achieve robustness. So this would be very different if we had built, like some of our colleagues, all our architectures based on transients. The next topic I'll be talking about is sequence generation, and there are spiky neural networks models, for instance, where the processing is just one large transient going through that system. That would not have that robustness property. And our challenge would be, we will tell those guys, your architectures don't scale. They don't actually allow you to link to the real world because of that lack of scaling.